Hi class, in this lecture we are going to be looking at the regulation of gene expression. The first thing we are going to be looking at is how the genes are regulated in bacteria. So we are going to find out one of the things called an APRA. Bacteria like to organize genes into multiple sets. So instead of making transcript for each gene, it can put multiple genes together into one transcript. That means that you make only one mRNA, but you have many genes on it. So you can make all those products at once. What is nice about that is they can control it. They can regulate those genes by either turning them on or turning them off. When they do not need them, we'll explain the different situation. The first one is the lactose operon. It will be turned on when the lactose present. The other one is the arginine operon. It gets turned off when arginine is present. So they are slightly different. One is induced, the other one is repressed. The first thing we are going to look at is the regulation of protein synthesis. In the previous lectures, we talked about how proteins were made through transcription and translation. Now, we want to be able to turn on our proteins when we need them and shut them off when we do not need them. In prokaryotes, we can regulate these genes when they are only required, and this is coordinated by operons. Operons are to describe having genes on one transcript. So it is a set of genes. So what's nice about bacteria is that they can turn on multiple genes simultaneously, which is the benefit of having an operon. When we talk about the operons, we have two types. We have the inducible, which means it is turned on. So when you do something, you turn it on. Again, so in this situation, we had catabolic operons, or enzymes need to be metabolized a nutrient or produced when it is needed. A great example is lactose. Most of the time, bacteria will prefer glucose to do cell respiration, but it can also do lactose and other carbohydrates when found. So when those things are found, they can turn on the machinery. Then, when the enzymes or the substrates are not there anymore, they can shut them off. So that is a way of turning on the enzymes when the substrate is present, then turn them off again when the substrate is not there anymore. So that is an inducible operon. I'll just show you examples of that in just a minute. The second type is the repressible. This is when you want to shut it off or when you do not need it. Usually, these genes are on. You turn them off because you don't need them to be on at that time. Typically, in this situation, you need to have the substrate all the time. So you turn those genes on all the time. Then when you come into an environment where the substrate is present, you do not need to make the genes, then shut them off. That is the idea of a responsible operon. So let us take a look at these operons and give you some examples of both types of them. So the first operon is called the lactose operon, an inducible operon. It has three segments. One, regulator, which is the gene that codes for the repressor protein, and this could be downstream of another DNA. Second, control locus, which has promoter and operator. This is where the repressor will bind. Third, structural locus. In lactose genes, the structural locus is made of three genes. Each gene codes for an enzyme needed by catabolize lactose. Beta-galactosidase hydrolyzes lactose. Perminase brings lactose across cell membrane. Beta-galactosidase transacetylase uncertain function. They are important for the different function of breaking down lactose. I do not mind if you know the enzymes and their functions or not. What I want you to do is to understand what does induction means. Usually, the lactose operons is off in the absence of lactose. This is because the repressor protein binds with the operator locus to prevent the RNA polymerase from binding to the DNA and blocks the transcription of downstream structural genes. Only when lactose is present, lactose will bind the repressor and pull it off the DNA. Then the RNA polymerase will start to work. Lactose turns the operon on by acting as the inducer. When lactose is present, lactose can bind to its repressor. The repressor protein changes its shape and causes it to fall off the operator. RNA polymerase can bind to the promoter. Structural genes are transcribed. Now it has the proteins to break down the lactose. Once all the lactose is gone, the lactose will fall off. Repressor will come back, bind on the DNA, and shut it off. Essentially, what happens is that lactose is what turns on the switch. Lactose is not there, the switch is off. Lactose is there, the switch is on. We call this a lac operon. This is the first operon discovered back in the early 60s. It figured out how genes are regulated in bacteria. There are also other operons, which are typically on. However, it shuts off when the process is unneeded. Arginine is an amino acid that is needed in lots of different proteins, so arginine is required by the cell. So it always needs to be present. When arginine is not in the medium, the bacteria have to make it. These structural genes are necessary to synthesize arginine, so arginine needed to be present. There is a repressor protein in the bacteria. So if we add excess arginine to the media, arginine will bind to the repressor and changes it. Then the repressor will bind to the operator and block RNA polymerase and stop the arginine synthesis. So arginine is the co-repressor. Alright, here is a little video to show you again both the repression and the induction. And then I will come back and talk a little bit more, and then we will move on.
When a microorganism, such as E. coli, is grown on glucose, it must synthesize all of its amino acids. If an amino acid, such as histidine, is added to the medium, the organism can turn off the synthesis of the enzymes needed to make histidine. In the absence of the amino acid, a regulatory protein called a repressor is made, but it is unable to bind to DNA and prevent transcription. When the amino acid is added to the medium, it enters the cell, reacts with the repressor, and activates it so that the repressor now binds to the operator site on the DNA and turns off the pathway by blocking RNA polymerase and preventing messenger RNA synthesis. When microorganisms such as E. coli are grown on glucose, they do not synthesize the enzymes needed to degrade other growth substrates such as lactose. In the absence of lactose, the regulatory protein, called a repressor, binds to the operator site and blocks transcription. When the substrate, which is called the inducer, becomes available, it reacts with the repressor and inactivates it. The repressor can no longer bind to the operator site and transcription can proceed. Regulation of catabolic pathways involving a repressor that is inactivated by the inducer is referred to as negative control. Some degradative pathways are regulated by an activator protein. An activator is a regulatory protein that facilitates transcription by binding to DNA. Activators must react with the substrate that they regulate, such as arabinose, before they can bind to the DNA. The binding of the activator to the activator binding site on DNA enhances the ability of RNA polymerase to bind to the promoter site. In the absence of the inducer, the activator cannot bind to the DNA. RNA polymerase is unable to bind to the promoter site and transcription does not occur. Regulation of degradative pathways involving an activator that is activated by an inducer is referred to as positive control.
Again, lactose operon usually runs off. When the lactose is present, it will turn the genes on, so it will be degraded. Arginine operon usually is on. When excess arginine is present, the genes will be shut off because you don't need to make it. So arginine operons get shut off by repression. Let us answer this question. If an operon's repressor is in its active form, that means transcription in the operon is occurring. or transcription of the operon is not occurring. If you answered that the transcription is not occurring, you are correct. When it's in an active form, that means it is bound to DNA. It no longer allows transcription to occur. Good job if you got it right.